Hi, everyone. My name is Kyle Lambert. I'm a graduating senior in the neurobiology track of the biology major, and I'm excited to talk to you about my project on regulating paraneoplastic neurologic syndrome autoantigens in small cell lung cancer. So just to give a brief overview, a lot of the recent work in cancer research is focused on the mechanisms, the genetic and molecular mechanisms behind cancer progression, metastasis, as well as these autoimmune responses that we see in some patients that I'll further characterize here. So small cell lung cancer specifically is extremely aggressive and has a poor prognosis, about a 5-year survival rate of 7%, constituting just under a quarter of all lung cancers worldwide, yet being the most lethal. These paraneoplastic neurologic syndromes, on the other hand, are characterized by this autoimmune response inducing neuronal damage that often occurs in SCLC patients. And what's actually happening here is that the patient's cancer cells are expressing these neuronal proteins that are exclusively found in brain tissue, nervous system tissue, and nowhere else in the body. The body then recognizes that as a foreign entity. The immune cells then start attacking not only the tumor, but also the body's normal, natural cells that are meant to express those neuronal proteins. Uh, so what I was interested in here was looking at this driver of neuronal programs in small cell lung cancer, NF1B. It's a transcription factor that has been previously characterized as driving neuronal programs as well as metastatic programs in small cell lung cancer. So what I wanted to look into here was whether tumors that express high NF1B would have these increased PNS-related antigens. And here I have a bit of a summary of what these antigens are. I focus specifically on the Hu antigens, Hu R, B, C, and D, with Hu C and Hu D being two SCLC PNS-specific antigens. So these are the antigens that are used for antibody testing in the clinic to diagnose a patient with paraneoplastic neurologic syndromes associated with SCLC. So first and foremost, the large finding that I had here was that NF1B high neuroendocrine cell lines had higher neuronal hue expression. So I looked into this in a plethora of ways. One, I looked into the expression itself of the Hu antigens in NF1B high cell lines versus NF1B low cell lines, as shown in the heat map. Above, you can see that there is this elevated expression of Hu antigens in the NF1B high cell lines. I also performed a qPCR assay that showed significant decrease in neuronal Hu gene expressions between NF1B high cell lines and NF1B low human cell lines. Uh, to further this and to solidify my findings in protein setting, I performed a quantitative immunoassay through WES that showed an elevated expression of these neuronal Hu proteins in NF1B high cell lines compared to NF1B low cell lines. Um, I also decided to stratify the distribution of Hu expression levels based on SCLC subtypes. So there are 4 different subtypes of SCLC that have previously been characterized based on the gene expressions of ASCL1, NeuroD1, YAP1, and P. So there are 4 subtypes, A, Y, N, and P, with A and N both being the neuroendocrine type. What I found here was that the Hu antigens are almost exclusively expressed in these neuroendocrine cell lines with very minimal amounts found in those that had higher expression of non-neuroendocrine. So to further look into the actual mechanism by which NF1B could actually be increasing expression of these neuronal Hu antigens, what I looked into was ChIP-seq and ATAC-seq data, which showed chromatin immunoprecipitation and where there's actual binding of this transcription factor in a mouse genome. And what I saw here was that NF1B actually binds upstream of the transcription site of ELAV-L2. So ELAV-L2 is a gene that codes for HuB. While HuB is a neuronal Hu protein, it has not been characterized as one that's particularly important for the diagnosis of SCLC PNS. So when patients go into the clinic for antibody testing, they're not being tested for antibodies for HuB, rather only for C and D. So this was particularly interesting because it pointed to a way in which NF1B could be regulating these SCLC-specific PNS antigens, perhaps mediated by ELAV-L2 or by its protein HuB. So to further look into this, I looked at log2 fold change of expression of HuB following the NF1B knockdown or overexpression in a NF1B high and NF1B low cell line, respectively. And what I saw here was that NF1B likely acts as an enhancer. When we knocked down NF1B, we saw a decrease in ELAB-L2. When we overexpressed NF1B, we saw an increase in E level 2 or HuB. So NF1B acting as an enhancer of E level 2 was particularly interesting because previous studies have shown that E level 2 has interactions with E level 3 or HuC that is positively regulatory. So that means by NF1B binding and enhancing E level 2, we're suggesting a possible mechanism by which there's this increase of these PNS-related antigens. So I was particularly excited about this particular conclusion, but moreover, that E level 2 might actually prime this neuronal spliceosome activity through HuC and HuD, guiding this PNS antigen presentation.